All righty, let's go ahead and get started. Good morning. This is going to be the lecture for Monday, April 19th, 2021. We are onto the second part of our urinary system where we have focused on the anatomy for the first lecture. And now we're going to start talking about how it's regulated and the processes and everything that goes along with that. We still have a couple more assignments due on Wednesday, the 21st, one more unit review and one more Physio X that does have 6 activities. So make sure you give yourself time to do that. And then you got a whole long weekend to study both the respiratory and urinary system. If you think about it, we'll have done 6 lectures total, 3 of each. So that means both the lab and lecture exams are going to be about 50% on the respiratory system and 50% on the urinary system. So be prepared for both. As I mentioned, on Wednesday, the 28th, we start the reproductive system. I think we have 4 lectures for that. Am I saying that correctly? Yep, absolutely 4 lectures for that. And the first one, we're going to spend a fair amount of time talking about mitosis and meiosis. So making sure we're familiar with mitosis. It's been a very, very long time. Cells in 430 since we've talked about mitosis, to help everybody kind of remember what we need to focus on for that. There is a labster activity that you're going to do after the exam that's going to be due Wednesday, the 28th. And like all labsters, remember, you must complete the whole activity and get it at least 80% correct for full credit. All right, excellent. So let's go ahead and get started with the lecture. We left off last time and we had done all of the anatomy. And let's cheat a little bit on this, bring back up the whiteboard now that I've gone away from it. And simplify things a little bit. We know, of course, that the job of the kidney is to filter the blood. And to do that, basically, we have that special structure, that nephron, that is made up of basically 2 main components. We have the blood vessels. So, of course, we have the afferent arteriole, which is not a part of the nephron that feeds into the glomerulus. And then let's actually, now that I've done that, I want to cheat and undo that. The glomerulus. It then comes out and forms the efferent arteriole and the peritubular capillaries and vasa recta. So we'll do that there. So again, we've got, and we can put some little tiny labels on this. Glomerulus. And then the efferent arteriole. Which feeds into the peritubular capillaries. And could also include the vasa recta. And again, we have our quote unquote dirty blood. That enters into this capillary. Of course, the nephron also includes tubular structures. We can do those in blue. Starting with Bowman's capsule, which then feeds into the proximal convoluted tubule, distal convoluted tubule, and the loop of Henle. And I'm just going to abbreviate. Proximal convoluted tubule, the loop of Henle, the distal convoluted tubule. And even though it is not a part of the nephron, remember we did talk about how the collecting ducts will help to process the filtrate. So at the end of this process in our blood vessels, at the end of this, as it enters the venules and goes back into the veins, we have our now clean blood. Why did that not write? And of course, everything that stays in the tubule becomes urine. Excuse me. So this is the process of, that filtration and processing that takes place in the nephron. And this process, with this very simplified version, how we process and filter the blood basically involves three major steps. So what I want to do now is we can talk about these three major steps, where they occur, what they involve, and everything else involved in that. So I want to take this and divide this here into three, and we can talk about the three processes that are involved in the filtering of the blood. Now, the first of these is a process called glomerular filtration. As the name indicates, where do you think glomerular filtration takes place?
In the glomeria.
Yeah, there you go. Exactly. See how easy that is? Absolutely. It takes place in the glomerulus. It is filtration, so it is an outward movement. And notice in this case, when I'm saying outward, I am meaning out of the body, right? If you think about it, these tubules, because they ultimately connect to our urethra, to the outside of the body, basically anything that is in these tubules and stays in the tubules is going to leave the body. So this is basically moving outward, movement from the blood to the filtrate. And in fact, glomerular filtration is really where we make the filtrate. Obviously, this is going to only happen in the renal corpuscle. And basically, this is a passive process. It relies solely on fluid pressures. We know a little something about that because we talked about capillary exchange way back in the cardiovascular system. And we were able to talk about some of those fluid pressures. And if you remember, they were the hydrostatic pressures and the osmotic pressures. And we'll talk about those again if you don't recall them. But we definitely will talk about those again. But as we know, if something is passive, that means we are not using ATP for it. And while that is fine that it's not using ATP, the problem with this is that we cannot be specific in this process. We can't decide what comes in and what goes out. Basically, in this process, it is selective only by size. Anything that is big enough to come out of the glomerulus is going to come out of the glomerulus. And that means both good things and bad things. I always, in my own mind, I always equate this to, right, you go away for a week on holiday, and when you come back, you have that huge, massive stack of mail. So the very first thing you do is separate the mail by size, because most of the big stuff are the mailers or the advertisements or things along those lines. It's the junk mail. And most of the small stuff are the important stuff, like the bills and things along those lines, right? But of course, the key word there is most. Is all of the small envelopes things that are necessarily going to be important and things that you need and must have? No, of course not, right? There's going to be some junk mail in that as well as, right, some of those big letters may be your acceptance letter to Stanford or something along those lines. So again, we're being selective by size. So both good, and again, I'm going to use quotes for this, and bad stuff exit the blood into the tubule. It's fine if the bad stuff leaves, because again, once it's in the tubule, we can get rid of it. However, all that good stuff, like sodium, like amino acids, things along those lines, are things that we want to get back in. All right. And this absolutely positively is step one in the process. The first thing that happens is this glomerular filtration. All right. Questions on that? All right, excellent. So then let's talk about the second step in this process. Second step in this process is what we call tubular reabsorption. With tubular reabsorption, as the name would indicate, this is an inward movement. I misspelled from. From the tubule, or let's say from the filtrate, to be consistent, back into the blood. Excellent. All right. This process mostly occurs in the proximal convoluted tubule. But again, key word there is mostly. It also occurs in the loop of Henle, distal convoluted tubule, and collecting duct. Now, with this tubular reabsorption, what type of stuff are we going to want to bring back into the blood?
Water?
True, absolutely. Definitely water is going to be one of them. Absolutely. Water is definitely one of the things that we want to bring back. And what else?
Glucose?
Yeah, all that good stuff.
Not good. Salt?
Exactly. All the quote unquote good stuff we were talking about. Things like you said, things like, get that up. Things like glucose, things like amino acids, things like sodium, all the quote unquote good stuff that we want to bring back. Now, notice in this case, we want to be selective. We don't want just anything to be able to come back in. So this is going to be selective. This is going to be specific. However, to make this process specific, what is it going to require that filtration didn't? Right? We could just poke holes in something and let whatever fits through those holes out. And that could be passive. We don't have to be selective. We don't have to exert any energy to do that. However, if we want to inspect something and make sure that's something that we want that bring back, that's going to require energy. So this is an active process. It requires ATP. To be selective, to be specific in what comes back, we need to be able to use ATP to be able to do that. All right. Questions on that? All right. That brings us to the third process. The third process is tubular secretion. As the name indicates, this is again an outward movement from the blood into the tubule. Now, at this point, notice I didn't put a number here like I did the first one, because obviously glomerular filtration has to occur first. Let's do this. Glomerular filtration, as we talked about, is that movement of materials out of the glomerulus and into the tubules. That is our glomerular filtration. We talked about tubular absorption, and for simplicity's sake, let's go ahead and make this purple to help us to remember. As we talked about, most of that movement back into the blood is going to occur in the proximal convoluted tubules, but some of it's going to happen, oops, wrong direction. Some of it happens in the loop of Henle, some of it happens in the distal convoluted tubule, some of it happens in the collecting duct as well. All right, so again, for consistency, let's make sure we know. So this was the black arrow, that was the purple arrow, and let's go ahead and use green arrows for our tubular secretion. It turns out with tubular secretion, it also mostly occurs in the proximal convoluted tubule. That outward movement is occurring in the proximal convoluted tubule. So notice it isn't truly a sequential process. It's not like all tubular reabsorption occurs first and then all tubular secretion occurs. They're both occurring at the same time, where again, most of the tubular secretion occurs in the proximal convoluted tubule. Some of it occurs in the loop of Henle. Some of it occurs in the distal convoluted tubule. Some of it occurs in the collecting ducts. So like tubular secretion, this mostly occurs in the proximal convoluted tubule. But also occurs in the loop of Henle, distal convoluted tubule, and collecting duct. Excellent. So really, this is, here, let's totally cheat. 2A, and this is 2B. So it really is kind of a 2.5-step process. Obviously, glomerular filtration has to occur first, but once that glomerular filtration occurs, then tubular secretion and tubular reabsorption are basically occurring at the same time. Now, we'll talk about them separately. We're going to talk about each process independently, but it's important to know that both of them are occurring at essentially the same time, and most of it is occurring in the proximal convoluted tubule. Of course, in this case, we are moving things out of the blood. And so those things that we're going to move out of the blood is bad stuff. This can be a combination of large wastes that weren't able to fit out in the glomerulus. There are actually, during reabsorption, there are some quote unquote bad things that sneak back in. We can kick out. This is going to include toxins that can be removed here, and substances that we want to remove based on our hormonal needs. So for instance, if we have too much water or if we have too much sodium in the blood, again, we have hormones that tell the kidneys to release that stuff, and this would be where that's happening as well. Again, notice we want to be selective. We want to be specific. And if we're going to be specific, that makes this an active process. And if it's an active process, it is going to require ATP. And so then after all three of these processes are completed, we then have our clean blood that goes back to our heart and our urine, which leaves the kidney into the urinary tract. So we've done this here on the board. Let's go back to the slides and see that same type of thing. Here we see this simple illustration for this 3-step process. Glomerular filtration happens only in the renal corpuscle. Outward movement from the blood into the tubules, forming the filtrate. And that filtrate is basically water and all of the little stuff that fit through that fenestrated capillary and that those filtration slits formed by the podocytes. Tubular reabsorption is the active and selective process of moving good stuff back into the blood, things we want to retain. Remember, as we talked about, we produce here in glomerular filtration, there we go, 200 liters of filtrate in a 24-hour period of time. So obviously the majority of that, vast majority, over 99% of that needs to be reabsorbed back into the blood supply. But there's still going to be some stuff in that blood we need to get out as well. So again, we've got it as a 1, 2, 3-step process, but remember, tubular reabsorption and tubular secretion are basically occurring at the same time. That is a good question. I don't think I saved the drawing, so let's go ahead and do that. There you go. I saved it now. Thank you for asking that. All right, excellent. So let's work our way through these processes, starting with glomerular filtration first. Again, as we've talked about, this occurs only at the renal corpuscle. And again, as I keep emphasizing, filtrate is very, very different from urine. Aside from the fact that we are producing 200 liters of it, and we don't produce 200 liters of urine, essentially what filtrate is blood plasma and anything small enough to fit through that filtration membrane. Right? So really the big difference between blood plasma and filtrate when it is first produced is that the filtrate doesn't have anything big, right? Remember, this membrane basically divides by size. So anything big, large proteins, formed elements, right? You typically don't have blood cells, white, red, or platelets even in your urine. Right, so basically just divides things by size. Anything large, all the formed elements all stay in the blood. And remember, as we've talked about, it is not selective. Anything that is small, good and bad, comes out of the blood plasma. And that's why we need that tubular reabsorption process to get all that good stuff back in. Conversely, the 1 to 2 liters of urine that leave your body during a 24-hour period of time is primarily metabolic wastes, any substances that we don't need, and a small amount of water. All right, excellent. Now, again, this is a passive process, just like gas exchange. So again, there are some ways we are going to want to maximize the efficiency of this. One is it is primarily going to be completed by the pressures of the capillary. Remember, one of the things that we talked about, one of the special things about the glomerulus is the glomerulus has a high pressure to it. And that blood pressure is going to be what forces the filtrate out. As the blood passes through the glomerulus, about 10% of the plasma comes out as it passes through. So like I said, during the course of the day, that produces about 200 liters of filtrate, which we then reabsorb over 99% of to make those 1 to 2 liters of urine during the course of the day. To maximize the efficiency of this, remember, we have this thin membrane. As you can see from this illustration, I've got a great light microscopy view that I really like of this as well. But basically, as we know, it is a simple squamous endothelium of our capillary, which, as you can see, has those fenestrations, those pores in it that makes it more. We also talked about how we have those foot-like processes of the podocytes, right, that come together, and then a little bit of basement membrane that holds them together. So basically anything that can fit through the filtration slits and the fenestrations are going to be able to sneak their way out. Each kidney has over 1 million nephrons, so we have a huge surface area for this. And again, it's a very short distance that it has to travel. And then that high glomerular blood pressure. So these are the ways that we are maximizing the efficiency of this process. Now, I keep talking about that high glomerular blood pressure, but we know how capillary filtration takes place and reabsorption takes place. So we can actually talk about that. Here's the pretty picture that I mentioned. Again, we've talked about the filtration membrane already. Here is our fenestrated capillary, just a simple squamous epithelial tissue with holes in it. the visceral layer of Bowman's capsule, which are those podocytes with the foot-like processes, and that fused basement membrane or basal lamina that holds the two together. This is known as the dense layer, but again, it's just the basement membranes that hold them together. Here's an illustration like we saw in the textbook, but here is this great electron microscopy view where we can see this. Notice here we have our simple squamous epithelium with those fenestrations. Here we see the foot-like processes of the podocytes. Here we see their basement membranes holding them together. And we can see that basically something would have to sneak through one of these fenestrations, through these filtration slits to be able to get out. And as it said, and we'll cheat and go back to the previous slide, this gap is only about 6 to 9 nanometers wide. So again, no formed elements, no large proteins are sneaking out, just all the little stuff, right? Ions, metabolic wastes, metabolic materials like glucose, amino acids, things along those lines as well are all sneaking out. So anything small in the plasma and some of the water of the plasma, about 10%, leaves in the glomerulus. Now, we know it is a filtration pressure because we are able to calculate those pressures. Now, if you remember correctly, let's go back to our whiteboard, and I did save this, so thank you for reminding me of that. Let's take a look at our capillary, and we'll change our drawing a little bit. So here is our glomerulus. Oh, hold on. I'm just going to draw the glomerulus as a big capillary. Rather than being all squiggly, we'll draw it this way. And of course, as we know, Bowman's capsule, and I'll just draw Bowman's capsule like this, and there's our proximal convoluted tubule, where it collects it as well. If you remember when we talked about capillary exchange way back in the cardiovascular system, we talked about the hydrostatic pressure of the capillary. Right, and we talked, and that was the force of the blood pushing on its boundaries. Essentially, it is the blood pressure. And then we also talked about the osmotic pressure of the capillary. Right, this was basically the stuff in the blood that attracts water. Right, any of this sounding vaguely familiar, hopefully? All right, we also talked about interstitial fluid has hydrostatic pressure and osmotic pressure as well. Now notice we don't have interstitial here, but this is Bowman's capsule. And in this capsule, there will be some pressures in them. Now, first of all, obviously the capsule wraps very, very tightly and closely against the capillary. So let's say, for instance, you have a hose and you turn that hose on and you take that hose and put it all the way down so it's super close to the bottom of the bucket and you turn it off full bore. What happens when you do that, when you put it right next to the end of the bucket? Anyone ever done that before? Anybody awake? Want to even type an answer? All right. It bounces off of the bucket and kind of sprays all over the place. So absolutely here in Bowman's capsule, there is going to be a hydrostatic pressure. There will be a hydrostatic pressure here inside of Bowman's capsule. But Osmotic pressure involves the stuff in the filtrate being able to attract water to it. Remember here, when we're making the filtrate does not stay in the capillary, in the capsule. Instead, it leaves out the proximal convoluted tubule. As a result of that, the water and the stuff in the water doesn't stay around to attract water to it. So here in our glomerulus, there is no osmotic pressure of the capillary. So notice when we are determining our net filtration pressure, for that net filtration pressure, really there are just 3 pressures we have to worry about. Now, remember, one other thing we learned about with these pressures is there are outward pressures and inward pressures, right? Ones that go out of the capillaries, ones that come into the capillaries. Our hydrostatic pressure of the capillary is going to be an outward pressure going out of the capillary. Osmotic pressure is going to be an inward pressure because it draws water in. And our hydrostatic pressure of Bowman's capsule is going to be an inward pressure as well. So 2 of these are going to be trying to push water and stuff into the capillary. And one of them is going to be an outward force. Of course, we figured out filtration pressures, net filtration pressures before. So to calculate our net filtration pressure, basically, we just need to calculate the outward forces minus the inward forces. So in this case, our net filtration pressure would equal the hydrostatic pressure of the capillary minus the 2 inward forces, the osmotic pressure of the capillary plus the hydrostatic pressure of Bowman's capsule. And if we could plug and chug those numbers, we would then be able to calculate what the net filtration pressure is. So let's take a look at this. Starting first with our glomerular hydrostatic pressure. Does anybody remember what we said the pressures were in a typical capillary way back in the cardiovascular system? We noticed we had 2. There was at the arterial end and at the venous end. Venous. Does anybody remember what those 2 pressures were, the hydrostatic pressures? Yeah, exactly. It was, well, not 25, 26, close. It was 35 at the arterial end, and it was 17 at the venous end. So if you notice, our glomerular hydrostatic pressure is almost double what the pressure was in a typical capillary at the arterial end, and almost 3 times what it is at the venous end. Remember, one of the things we talked about, one of the specializations of the glomerulus is that it is very high pressure. So we have this huge high pressure of 55 millimeters of mercury with that outward force. Our capsular hydrostatic pressure, the pressure inside of Bowman's capsule, is 15 millimeters of mercury, forcing water back in. And then, like we talked about, that osmotic pressure of the blood inside the capillary is 30. So let's add these numbers to our picture. Here we know this outward force is 55 millimeters of mercury. Here we know this inward force is 30 millimeters of mercury. And we know this inward force is 15 millimeters of mercury. So if we come back up here to our equation and do a quick plug and chug, our net filtration pressure is equal to 55 minus 30. Let's put 30 plus 15. So what does that work out to? What is our net filtration pressure equal? Come on, you shouldn't have to take off your, there you go. A positive 10 millimeters of mercury. Excellent. Obviously, being a positive force, we know it is an outward force of 10 millimeters of mercury. So the net effect of this is we get that outward push of blood, of plasma really, and all the small stuff in our plasma out of the glomerulus. And again, we see that, we did that, and here's the pretty picture associated with that as well. All right. Questions on this? All right, let me ask you a quick question. If I go from laying down to standing up and my systemic blood pressure drops 5 millimeters of mercury, is there a dramatic change in how blood is going to be pushed through my body from the tip of my toes to the top of my head with just a 5 millimeter of mercury change in my blood pressure? What do you think? If my blood pressure suddenly goes from 120 over 80 to 125 over 80, is that a significant difference where I need to be rushed to the hospital to make sure that everything is going to be okay? No, those types of changes are minimal, absolutely. However, here in the glomerulus, if we have a 5 millimeter of mercury change, is that going to affect the function of my glomerulus and my ability to be able to filter my blood? Yeah, it's going to have a huge effect. Absolutely. So maintaining a consistent glomerular filtration pressure is going to be vitally important, because here, even small changes of pressure can have a huge impact on function. So while it is important to be able to calculate this net filtration pressure, we have to make sure we understand how to control it as well, because even small subtle changes in this pressure can have a huge impact on our ability to be able to filter our blood. Now, the force in the glomerulus, that net filtration force, determines, and let's actually write that because it doesn't say it here. Our net filtration pressure determines the glomerular filtration rate. Basically, how much stuff how much filtrate is formed, how much comes out of the capillary into Bowman's capsules. In males, it's about 125 millimeters of mercury. In females, it's about 105. Again, these are statistically significant because, again, of the huge N that we're dealing. However, again, from a functional standpoint, is there a significant difference here? No, of course not. I know Allison thinks that males are better, but in fact, these differences mostly have to do with size. Males are slightly bigger than females, and so they get a slightly larger number. But obviously, There's huge variations in this between people. See, she agreed with me. Excellent. But as we talked about, it is vitally important to maintain appropriate rates of this. Remember, we were talking about this in the last class. If our glomerular filtration rate is too high, what ends up happening is we make too much filtrate. And if we make too much filtrate, remember, we don't have time to process it properly. And so what happens is our filtrate is under processed. Conversely, if our glomerular filtration rate is too low, we don't make enough. We make too little filtrate. And remember, if we make too little filtrate, then our filtrate is going to be over processed. So it is really important to maintain the appropriate rates of this pressure, which determines our glomerular filtration rate. So not surprisingly, we have three main ways that we need to control and regulate this process. And not surprisingly, it is going to involve both of our communication specialized organ systems, nervous system and endocrine system. So we are going to use both nervous and endocrine. Oops, that's not how you spell involve. Nervous and endocrine systems. And notice I said three methods because under nervous, we are going to have both intrinsic, contained within the organ itself, and extrinsic, which of course means going through our central nervous system, right? Going through our autonomic nervous system, as we know, for these types of organs. All right. So questions on our baseline of what needs to happen here in our glomerulus to make our filtrate. Any questions on this? All right. What we need to do now then, like I said, is we need to talk about how we are going to regulate the rate of glomerular filtration. Now, again, we are talking about the movement of water, right? Fluid, let's just say fluid. And the pressures involved in that. When we talk about this, there are two concepts we need to think about and understand. Two related but independent concepts related to this. The first involves the flow. Let's make this capital. Of fluid. All right. So obviously, how do we want to do this? The more blood that flows into the glomerulus, the more filtrate we will make. Does that make sense? Yeah. So notice in this case, if we were to dilate a blood vessel, as a result of that, we would get increased flow. And with more water, more blood, we get more filtration. Okay? That makes sense? Conversely, the opposite is true. We constrict a blood vessel, that is going to decrease flow. So less blood. And then as a result of that, we get less filtration. I think that part makes sense. That part is easy. But here's the problem with this. When we change the diameter of the blood vessel, not only do we change the flow of the blood, the amount of blood that flows in there, but we also change the pressure of the fluid, of the blood, right? Because as we've talked about, blood pressure is the force of the blood pushing against the walls of the blood vessel. All right, that makes sense as well, hopefully. So here's the trick. Notice when we dilate a blood vessel, what does that do to the pressure of the blood inside of it? Right? If we dilate a blood vessel, there's more volume, more space, let's say it that way, in the blood vessel. And when there's more space in the blood vessel, what happens to the pressure of the blood? It decreases. Excellent. Pressure in a blood vessel decreases. Now, if that pressure in the if the pressure decreases in the glomerulus, what's going to happen to our filtration rate? If we have a lower pressure in our blood vessel, are we going to get more filtrate or less filtrate? Yeah. Conversely, if we constrict a blood vessel, there is less space in the blood vessel. And as a result of that, pressure in the blood vessel increases. And if we had an increase in pressure in the glomerulus, we could produce more filtrate. Notice these two concepts seem to be at odds with each other. When we're talking about flow, increasing the diameter of the blood vessel gives us more filtration. But when we're talking about pressure, increasing the size of the blood vessel gives us less pressure. So we do have two kind of dynamically opposed processes here. And so it's important to remember when we're talking about these changes in diameter of the blood vessels we're going to be discussing, we want to think about whether it is affecting how much blood enters the glomerulus, or we want to think about if it's affecting the pressure of the blood. Oops. Because depending on which one of these two factors it is influencing is going to determine what type of effect it's going to have on our glomerular filtration rate. I appreciate on the surface this looks a little confusing, but I'll try to walk you through it as easily and as slowly as possible to make sure that this makes sense. So let's go ahead and start. What do I want to do? Let's go back to the lecture. All right. We are first going to talk about our neural controls. And with our neural controls of glomerular filtration rate, there are two types: intrinsic and extrinsic. Of course, as we know very, very well, intrinsic refers to within the organ itself, whereas extrinsic means that it is going through the central nervous system. And because we're talking about the urinary system, it's coming out the autonomic. Again, shouldn't be any surprises to that. So let's talk about the intrinsic mechanisms first. And there are two intrinsic renal autoregulatory processes. The first is what we call a myogenic mechanism. This myogenic mechanism basically is going to allow us to respond to dynamic changes in systemic blood pressure. All right. As we've talked about, when you stand versus when you sit, when you lay down versus standing, when you're walking versus sitting still, while you're eating a lunch, while you're getting surprised by something that you're watching on TV, while you're laughing, right? All of these different things are causing dynamic changes in your blood pressure. Small changes in your blood pressure, right? 1 to 5, 1 to 6, 1 to 10, millimeters of mercury changes, which when we're talking about systemically through the whole body, don't make that big of a difference. But like we talked about, even a 2 or 3 millimeter of mercury change can have a 10 to 20% effect on our glomerular filtration rate. And we don't want that to occur. So notice with this myogenic mechanism, our goal here is to regulate, or let's say it this way, maintain constant pressure, and let's make this in big letters, in the glomerulus. So our goal is to maintain a constant pressure in the glomerulus. Right? Notice we're not talking about flow here. In this case, we're talking about pressure. So that can help us to understand what's going to happen. So let's think about what happens. Oh, and I guess there's one more key point I have to mention about this. This is going to help us maintain constant pressure in the glomerulus. And this myogenic mechanism occurs in the afferent arteriole. So let's think about our first possible imbalance. That first possible imbalance is there is an increase in pressure in the afferent arteriole. So the pressure in your afferent arteriole goes up. That means the blood coming into our glomerulus would be at a higher pressure, and that would cause an increase in filtration rate, which we don't want. Now, we are able to recognize this because there are stretch receptors in the afferent arteriole. So when pressure goes up, the afferent arteriole, and I'll just abbreviate that AA now, stretches. And using intrinsic controls, how do you think our afferent arteriole responds? How could the afferent arteriole bring the pressure back down? Come on, give it to me. Pressure inside of a blood vessel increases. How can a blood vessel bring that pressure down? Will it bring the pressure down by constricting, or will it bring the pressure down by dilating?
Dilate.
Dilating, exactly. And so what happens is our afferent arteriole responds by dilating. When it dilates, it lowers the blood pressure and maintains the constant pressure we want. Bingo. Notice here our changes are involving pressure. And so we need to respond appropriately. So then hopefully this becomes even easier this time. Our imbalance now is a drop in pressure in the afferent arteriole. Right? We have stretch receptors that now fire less. So when the pressure decreases, our afferent arteriole rebounds. Let's say it this way. Stretches less. And when it stretches less, our afferent arteriole is going to respond by doing what? If our afferent arteriole wants to bring the pressure up, what does it need to do? Constrict or dilate?
Constrict.
Constrict. Excellent. So notice what's happening in our afferent arteriole is it is constantly constricting and dilating to try to maintain a consistent pressure of that blood as it's entering into the glomerulus. Right? Notice this is not about the volume of blood. This is just about the pressure. And that is what this myogenic. Remember, as we were talking about before, there are two factors that are going to include our glomerular filtration rate. The flow of blood coming into the glomerulus, but also the pressure of that blood coming into the glomerulus. So this myogenic mechanism is all about maintaining constant pressure. All right. Questions on this one? All right, excellent. If we understand this one, then the good news is we already learned the second one. The second regulating process of the kidney, that intrinsic one that we talked about, is that tubuloglomerular feedback. Right, changes in the juxtaglomerular apparatus. Now for this one, and let's go back because I want a little bit more room to write than we've got here on the board now. So we can come back here. Let's go ahead and erase this and remind ourselves of what's happening in that juxtaglomerular apparatus. We talked about this in the last class, but again, we talked about a lot of stuff in the last class, and it's always important to go over things again. Remember, this includes three types of cells. The first of those three types of cells are the macula densa cells of the distal convoluted tubule. The granular cells are juxtaglomerular cells. Of the afferent arteriole. And the mesangial cells that are, they're not in the glomerulus, but let's say under. Right, kind of embedded between the loops of the glomerulus. Now, our myogenic intrinsic mechanism that we talked about was all about regulating pressure. In this case, our goal is to regulate the flow of the water, of the blood, and really also regulate the flow of the blood and the surface area, right? Basically, how much blood is in contact with that filtration membrane. Right, so that is our goal in this case. Now remember, we had two possible imbalances. And remember, these imbalances, we'll come back to what the imbalance is in just a minute. But remember, the process starts with those macula densa cells, which are sensing the osmolarity and chemicals in our urine, although it's not urine yet, almost urine, in our filtrate near the end of the process in the distal convoluted tubule to see if we're over processing or under processing. So in the case here, if our macula densa cells determine that the filtrate is under processed, what did that indicate was the imbalance?
Too much fluid?
Yeah, we are producing too much filtrate. So we need to decrease the amount of it. So the macula densa sends signals to our other two sets of cells, starting with our juxtaglomerular cells. And since I wrote it out once already, I can abbreviate our JG cells. And what did our JG cells do as a result of this? In our afferent arteriole, if we're producing too much filtrate and we want to change the flow, decrease the amount of blood that is going into the glomerulus, how can our afferent arteriole decrease the flow of blood into the glomerulus? How can it do that?
Constricts.
Yeah. So it constricts. All right. The macula densa cells also tell our mesangial cells to collapse. Right, we want them to get smaller because that will decrease the surface area. And if we decrease the surface area, that will reduce the amount of filtrate that's produced. And again, to get these cells smaller, are we building up actin or breaking down actin?
Breaking down.
Yeah. And when they break down that actin, they get smaller, they decrease the surface area, and we've then, with these two processes, accomplished our goal. Our goal in this case was to make less filtrate and bring it back into balance. I can bring that up there so I have room. Remember, it works the other way as well. Another possible imbalance that our macula densa cells could determine is that we are over processing. So what would the imbalance be in that case?
Too little filtrate.
Yeah. We're producing too little filtrate. So again, it is going to send signals. Oops. And we didn't mention it in the last one, but we'll mention it here. What is the signal that it sends, especially to the juxtaglomerular cells? Anybody remember? How do we get that blood vessel to dilate or constrict? What gas was released?
Nitric oxide.
Yeah, that nitric oxide. Excellent. So again, it is going to send a signal to the JG cells of the afferent arteriole. And in this case, we want to increase the flow. Again, we're not talking about pressure this time. We're talking about flow of the blood into the glomerulus. So it's going to do that, in this case, by dilating. And we want to do the same thing with the mesangial cells. They are going to build up axon. When they build up actin, the cells get bigger. When the cells get bigger, they push up on the glomerulus, and we increase the surface area of the glomerulus. And when we increase the surface area of the glomerulus, we produce more filtrate. Right, and that, of course, is our goal. Make more filtrate because we are not making enough. We are over-processing it. Again, this isn't new. We talked about this at the end of the last class, but I know we were all tired by the end of the last class. So I think it's worthwhile to talk about these two things again. So notice the beauty of both of these systems, these intrinsic systems, is that every single one involves the afferent arteriole feeding into a single glomerulus. So every single 1 million of those nephrons in each of your kidneys is able to, just by itself, by measuring the pressure of the blood coming in, by influencing the flow of the blood coming in, maintain an efficient glomerular net filtration pressure so that it can set an efficient glomerular filtration rate. So every single one of those 1 million may be working at slightly different rates, but they're all going to be working at their maximal efficiency to effectively filter your blood. Again, we wouldn't want to have the oversight control to have to, consciously or even subconsciously for that matter, control every single 1 million of those nephrons per kidney. So the beauty of these intrinsic systems is it can handle it all by itself. Now, that being said, that doesn't mean we don't want some oversight control. And so we are going to have that oversight control, and that is going to come in the form of our extrinsic neural control and our hormones. All right, go ahead.
The process we just went over again, that's part of part B, the tubulo-glomerular feedback.
Right, exactly, because it's in the name. It tells you everything about it. It's between the tubes, primarily the distal convoluted tubule, where we have the macula densa cells. Right, and the glomerulus, which is basically right where the filtration takes place. So yeah, this basically tubular glomerular filtration, tubular glomerular feedback, right? Because we've got those quality controls in the macula densa that are telling the glomerulus what to do, and that uses those three regulatory cells. Oops, those three regulatory cells we talked about that are the juxtaglomerular apparatus. Bingo. All right. So we did that. We did that. All right. Questions on this? All right. So, as promised, way back in 430, when we first talked about our autonomic nervous system and how most organs get dual innervation, but the kidneys aren't one of them, we've had this weird concept that we've talked about that doesn't seem intuitive of how the sympathetic nervous system is what is primarily responsible for controlling the activity of the kidney. And ever since then, the very end of 430, I've been promising the opportunity to explain to you exactly how this works. And so let's do this. Let's start by going back to our whiteboard. Go ahead and clearing this off. We are talking about, and we can just rather than, we'll say autonomic control. But really, we know it's sympathetic. Of glomerular filtration rate. All right, and let's draw a little anatomy. Where do I want to do this? Okay, I'll do it twice. All right. As I'm sure you can clearly tell from this incredibly elaborate and detailed drawing that I have done, you can clearly tell that this here is our afferent arteriole. This here is our glomerulus. And this, of course, is our efferent arteriole. And you can clearly see I've done two completely identical and equally articulated drawings of these. And I can't BS and type at the same time. Excellent. All right, let's see if this works. Actually, you know what? Crap. Now that I've drawn all this, I think I want to move it all. Let's move this all down here. I think that gives me a little bit more room to write. All right. Obviously, the afferent and efferent arteriole are capillaries, not pardon me, are arterioles. They have smooth muscle in their wall. And let's talk about now what's happening when the body is at rest. Like when you're sitting here calmly in the classroom right now as we talk. As we know, when the body is at rest, as we've talked about, as much as 25% of our blood is going to our kidneys. Of course, the effect of that is we have a huge volume of blood and at a really high pressure, right? So we have a huge volume of blood coming to the glomerulus. And because we have a huge volume of blood coming to that glomerulus, the huge volume of blood at that good pressure is going to help to maintain a high glomerular filtration rate. All right. So at rest, everything in our glomerulus is fine. However, as we've talked about, when the body is active, as a result of that, much less blood is going to the kidneys. With much less blood going to the kidneys because it's being redirected to other areas. Right? Like the muscles, like the lungs, right? So that because we're actively moving and we're running around. So we have a dramatic decrease in blood volume going to the glomerulus. Since our volume is going down, with less volume, that means there's going to be less pressure. Right? As we've talked about before, right? Pressure is the force against the walls. If I took half of you out of the classroom and then counted the number of times you guys were bouncing into the walls, you would be bouncing into the walls much, much less. So there's going to be less pressure. And with less pressure, the problem with that less pressure is that our glomerular filtration rate will drop. And that's a problem. We don't want our glomerular filtration rate to drop every time we stand up and we walk around. We don't want to have to be sitting still all the time just so that we can make sure we're filtering our blood. And so, of course, when you are active, you have an increase in our sympathetic output. And here in the kidney, our sympathetic nervous system communicates with both the afferent and the efferent arterioles. And it tells both to constrict. Let's think about why that would be advantageous. What's the advantage? Whoops, that's way too big now. Of constricting the afferent arteriole. Well, when the arteriole gets smaller in volume, what happens to the pressure of the blood inside of it? Increases. So notice, while we have less volume of blood coming in here, constricting the afferent arteriole is going to increase the pressure of the remaining blood. So we have less flow, right? There's less blood coming in here, but we can increase the pressure. And that is going to help to maintain the glomerular pressure by increasing the constricting the afferent arteriole. And what is the advantage of constricting the efferent arteriole? When you're driving on 80, coming home from San Francisco over the weekend, or any time during the day, heck, it could be 4 o'clock in the morning on a Wednesday. You're driving home from San Francisco, going a good 90 miles an hour because it's 4 o'clock in the morning on a Wednesday. And then you hit Davis. And what happens when you hit Davis?
Major-
Yeah, the traffic slows down. Why does it slow down? Because you go from 75 lanes on 80 down to 2. Notice in this case, by constricting the efferent arteriole, we reduce the flow of the blood out of the glomerulus. It causes that same thing that happens in Davis. It produces congestion. And after all, when we have congestion in a blood vessel like this, this congestion means that the blood stays in the glomerulus for longer. And if it stays in the glomerulus longer, it has more time to bounce on the walls. And so that congestion staying in there longer increases the blood pressure. And that increase in blood pressure. So notice both of these take-home message. Nope, hold on. I wanted that to stay there. Come here. The goal of both constrictions, both, is to help to maintain a high blood pressure, even though the blood volume drops. And in this fashion, even when we're up and we're walking around and you're doing your treadmill exercises or things along those lines, we can help to maintain an even blood pressure and consistent filtration rate with moderate activity. So again, walking up and down the stairs, right? Shopping at the grocery store, right? Walking your dog around the park, things along those lines. What happens if we work at a high level, high intensity? What about if we're in that high stress, high intensity activity? Oops. If we're fighting that bear with an axe and we have lots of blood going to the muscles and to the lungs and very, very little going to the kidneys, am I going to be able to constrict that afferent arteriole and efferent arteriole enough to compensate for what's going on during that high intensity activity? No. So as a result of that, during a high intensity activity, kidney function will decrease. However, if you think about it, if you are fighting a bear with an axe, the fact that you're making slightly less urine or that your blood may be slightly dirtier than it was before is probably the least of your concerns, right? However, you don't want to be fighting bear with an axes for 3 days straight, because if that high intensity activity continues for a prolonged period of time, it can lead to kidney problems and kidney malfunction and problems with your blood. But those types of high intensity activities, fighting the bear with an axe, doing those sprints, running that race, doing that triathlon, whatever it is, those things typically last for a relatively short period of time. So there's no long-term implications to that. So like I said, on the surface, it doesn't seem to make sense that it would be the sympathetic nervous system that's in control. But remember, at rest, our kidneys are getting all the blood that they need to do their job. So it doesn't really take parasympathetic input to tell it, hey, do your job. It's getting all the blood it needs, nice large volume, nice high pressure, and it's doing everything it needs. When we're active is when we're getting less pressure, when we're getting less blood, and that is what we have to compensate for. So that's why when we're active, our sympathetic nervous system, by constricting the afferent and efferent arterioles, help us to compensate for the lower pressure, for the lower amount of blood, to maintain the high pressure needed to be able to maintain normal filtration rates. So hopefully now that makes a little bit of sense. All right. Questions on that? All right, excellent. So again, our sympathetic nervous system adjusts our afferent and efferent arteriole. And it also, among other things, stimulates our granule cells, which of course are important because, as we know, our granule cells are what produce our hormones and our hormone-like proteins. Which of course is our third type of regulatory mechanism. We have already talked a little bit in this class about these. We have already talked about anti-diuretic hormone. There shouldn't be anything new we have to say about that, except for one minor thing. We have already talked about atrial natriuretic peptide. That should be something we don't necessarily have to talk too much about. And what I mentioned before when we were talking about our endocrine system is that we would talk a little bit more here in the urinary system about this renin, angiotensin, aldosterone system. So that we do need to talk a little bit more about. But let's remind ourselves of these other two first. Anti-diuretic hormone. Someone remind me where it is produced. I always love blank stares when we're in a cumulative class with a cumulative final exam. Well, I'll give you a hint. Notice I asked produced by and released by as different questions, which is probably an indication, if you remember back to our endocrine system, that these are not produced and released in the same location. So certainly our antidiuretic hormone, as was pointed out, is released by, well, no, is it produced by the anterior pituitary?
Hypothalamus.
It's produced by the hypothalamus. Excellent. And what actually releases it? It's part of the pituitary, but was it the anterior that releases it?
Posterior.
Posterior, there you go. Oh, actually, I just realized the time. I went longer than I anticipated. Excellent. I'll tell you what, even though we've started this, let's go ahead and take our first break here. I apologize for going long. Let's go ahead and take our first break. It's 9:30. Let's come back at 9:45. And at 9:45, we will restart. Sorry, I apologize for that. I didn't realize. See, got so excited about what we were talking about. Wasn't paying attention. Excellent. So we'll restart at 9:45 and I will start the recording again at that point. Thank you for your patience. I'll see you guys in 15 minutes. All righty, excellent. So apologize for that weird breaking point, but I had just noticed the time, so we are back on now. We were talking about our hormonal regulation, and a couple of them are already familiar with, our antidiuretic hormone, produced by the hypothalamus and released by the posterior pituitary. And what we know about it, let's actually go back for a second. What we know about antidiuretic hormone is, as we've talked about, talked about how it tells the kidneys to retain more water. And at the time, we talked about how that will increase blood volume. And of course, if you increase blood volume, you increase blood pressure. So all we haven't talked about is where this occurs and how this occurs. Where this occurs is primarily in the distal convoluted tubule and the collecting ducts. in our collecting duct, specifically the principal cells, and in our distal convoluted tubule, what ends up happening inside of this is these cells have vesicles. And on these vesicles is a special protein. And this special protein is called an aquaporin. In the presence of antidiuretic hormone, basically what happens is that these cells, the cells in the distal convoluted tubule, and the principal cells of the collecting duct, are going to take these vesicles and actually express that aquaporin on the surface. Oops, that's not an arrow. Are going to come to the surface and they are going to express that aquaporin. And as the name aquaporin indicates, it is a channel specifically for water. So basically, when these cells express the aquaporin, water is able to be more easily reabsorbed. When antidiuretic hormone is not present, then what ends up happening is these aquaporins are endocytosed, brought back inside, put back onto a vesicle, and then can be expressed again later. So we're able to dynamically express and remove these aquaporins from the surface of these cells based on the presence of antidiuretic hormone. And of course, the last part of this is that if we are reabsorbing more water, like we said, that increases blood volume, that increases blood pressure. But the other thing that it does is it means less water is released in our urine. So it lowers the volume of our urine and it concentrates the urine. So the urine has much more solutes in relation to the amount of water that we have. But again, these effects on the urine is more just what we see from the outside. It's not important. It's not like we have to have a particular concentration of urine. We need to have the appropriate blood volume and the appropriate blood pressure. And so how much urine we produce and how concentrated that urine is more of an indication of how hydrated you are. When you're hydrated appropriately, antidiuretic hormone levels are low, your volume of your urine tends to be larger and it tends to be more clear in color, more dilute. If you are not hydrating well, then your body releases that antidiuretic hormone. You draw back more water to maintain your appropriate blood volume and blood pressure, and you produce a much smaller volume of concentrated urine as a result, right? So the color of your urine is an indication of how hydrated you are as an individual. Excellent. And again, really, other than knowing now about these aquaporins and where this takes place, there isn't a lot of new information on there. Similar thing happens with our atrial natriuretic peptide. Remember, this is the one that is produced by the atria of the heart in response to a high blood pressure. I remember, as we talked about way back in the cardiovascular system, the blood, when it returns to the atria, should be at a blood pressure of about 0. If it is not, then the atria produce and release this atrial natriuretic peptide. And basically what that does, primarily again in the distal convoluted tubule, it has basically 3 main effects. Well, really 2 main effects and then one influence of that. The first is that it decreases the reabsorption of sodium. So less sodium is reabsorbed. And of course, as we know, where less sodium goes, what follows sodium all the time?
Water.
Water. So if less sodium comes back, then obviously less water goes back as well, allowing us to increase the volume of our urine, get rid of and lower our blood pressure. Notice it also is going to suppress our antidiuretic hormone. So we stop expressing, you can do that over here, aquaporins. So again, more water leaves, less water is reabsorbed. And remember, aldosterone also drives the reabsorption of sodium. So notice it both directly and indirectly stops the reabsorption of sodium. So again, where that sodium goes, water follows. So more sodium stays in the filtrate, more water stays in the filtrate, and less is reabsorbed. So we get more water lost, less volume to our blood, and it decreases our blood pressure. Again, not a lot of new information there. We had already talked about these and what they did. So what that leaves us then is with the big one. Renin, angiotensin, aldosterone system. Now, the trigger for this is obviously a decrease in blood pressure or volume that is measured in the afferent arteriole. Remember, we already just finished talking about how our afferent arteriole has those stretch receptors. So if the blood volume drops or the blood pressure drops, our afferent arteriole is going to know it. We know that those granular cells, the juxtaglomerular cells, are going to constrict as a result of that, right? So we want to be able to increase blood pressure. But these granular cells, remember, also have an endocrine function. And in this case, they are going to release, and I've said this term a couple times, but I don't think I've written it out yet. This is a hormone-like protein. Renin is not a hormone, but it is a protein that acts very much like a hormone. And that renin, like hormones, gets into the blood. One of the primary things renin does is it goes to and causes the activation of an enzyme called angiotensin 1. Angiotensin 1 is produced by the liver. But where renin actually goes is it goes to the lungs. Why? Because it goes to the lungs. And in the lungs, there is a special enzyme. That enzyme is activated by the renin. And in the lungs, angiotensin 1, which is inactive, is converted into angiotensin 2, which is the active hormone. So, of course, the question is, why doesn't the kidney just make angiotensin 2? I don't know. For whatever reason, this is the long, convoluted, wandering path that this occurs. Blood pressure drops in our kidneys, produces renin that goes to the lungs, where it activates an enzyme that was made in the liver, and activates an enzyme in the lungs that converts an inactive hormone produced in the liver into its active form. So for whatever reason, that is the convoluted path that this is going to take. But this, as I mentioned, is the active form. And this hormone, angiotensin 2, does a lot of stuff. So let's talk about some of the things that it does. The first thing it does is it constricts Systemic arterioles, we want to make sure we're talking about that. Arterioles pretty much everywhere in the body. Now, again, remember the trigger for this was a drop in either blood volume or pressure. So if our systemic arterioles constrict pretty much everywhere in the body, what effect is that going to have on our blood volume or our blood pressure? Right. It's going to increase our blood pressure. All right. It's going to increase our blood pressure. Excellent. In the kidneys, it is going to increase the reabsorption of sodium, chloride, right, and water. So obviously, if we're reabsorbing water, if we're reabsorbing sodium and chloride, then of course, as we know, water is going to follow those. So both directly and indirectly, it is leading to an increase in water reabsorption, which is going to increase our blood volume. Angiotensin 2 tells our adrenal cortex, right, specifically, if you remember, our zona glomerulosa, to release a glucocorticoid. And that glucocorticoid specifically is aldosterone. Aldosterone, remember, leads to sodium reabsorption. And of course, wherever sodium goes, water follows. So this will increase blood volume. And I think I need to make smaller to fit in there. It stimulates our hypothalamus to make us thirsty. What happens when you're thirsty? And somebody should be able to come up with the answer to that one. What do you do when you're thirsty? I'll give you a hint. That's right, you drink. When you drink water, obviously you absorb more water from the digestive system. And as a result of that, blood volume goes up. Excellent. So notice all of the effects of all of these. Oh, and as if that wasn't enough, I guess I have to sneak this up here and do that. Stimulates the release of antidiuretic hormone, which, as we also know, increases water absorption. In the distal convoluted tubule and collecting duct. And again, more water back increases blood volume. So notice the goal of all of these, again, it was triggered by a drop in blood volume, a drop in blood pressure. So this is going to bring blood volume up. And of course, that is going to bring blood pressure up, helping us to maintain an appropriate blood pressure so that our kidneys can continue to perform at their optimal rate. And if you don't like all the pretty words that go along with this, your book's got a nice, pretty picture that does a good job of showing all of this as well. All right. Questions on this? All right, excellent. And again, more pretty pictures showing how all of these controls work together. When our blood pressure drops, we're going to bring that blood pressure back up to maintain that appropriate glomerular filtration rate so that our kidneys can continue to function to filter our blood and keep us, well, quite frankly, alive. All righty. That is everything you ever wanted to know and more about the glomerular filtration rate. We have talked about the glomerular filtration rate, what it is, how to produce it, how to regulate it, how to control it, and everything else that goes along with that as well. What that is going to leave us with, how are we on time? We are doing good on time, and let's talk a little bit more. Once process 1 is out of the way, then remember we have those 2 processes, both tubular reabsorption and tubular secretion, that are going to occur together. But again, they are separate processes, similar, but separate in their directions that they flow. Right? So they're similar processes using similar mechanisms, but they go in different directions. Here we see some examples of some of the materials that are reabsorbed and some of the materials that are secreted. One of the interesting things you may note as you look at this list is there are certain things like potassium that is both reabsorbed and secreted, like calcium that is both reabsorbed and secreted. Right? That's because especially when we're talking about ions, the key to our ions is we want appropriate levels. As we know, potassium is something that typically there are low levels in the interstitial fluid. So while we don't want to get all of it, get rid of all of it, we need to make sure that its level is low. And remember, as we've also talked about many times, calcium makes cells do wonky things. So our range of values for an appropriate calcium level is very restrictive. Again, this is a number I don't expect you to memorize, but it was something we talked about way back in 430. Basically, the range of calcium in your blood is about 8.5 to 11 milligrams per deciliter. And again, those are not numbers you need to memorize, but the point that I wanted to emphasize by showing you that when I do is just how restrictive that range is. Because calcium makes cells do wonky things. So having too much of it is really, really bad. Having too little of it is really, really bad. So there are things like that, that again, it's about making the levels appropriate. Sometimes that means reabsorbing more of it. Sometimes it means getting rid of it. So those are things that can move easily in both directions. Notice on the other hand, things like sodium, things like chloride, things like magnesium, which we want in very high levels in the interstitial fluid, pretty much are all just reabsorbed. All of our organic metabolites, glucose, amino acids, small proteins, vitamins, all of those organic materials are things we want to reabsorb. Whereas most of our wastes, especially those nitrogen-based wastes, are all things that we want moving outward in this process. So again, a lot of things are moving in just one direction. And of course, there are other toxins, drugs, neurotransmitters, all sorts of other things like that that are moved out as well. So most of this movement is occurring one direction or another, but the same type of mechanisms, active and passive transports, that are going to be used. And remember, as we talked about, over 99% of all of the materials, all of the filtrate is reabsorbed. And of that, about a little under 70% is occurring in the proximal convoluted tubule. So that proximal convoluted tubule is where most of the reabsorption and secretion work is taking place. So let's talk a little bit about some of these mechanisms for moving the filtrate or the substances in the filtrate in and out, determining ultimately what stays in there and becomes urine, and what leaves and becomes, it was brought back into the body and becomes part of the blood, or what stays in the filtrate and ultimately becomes urine. As I mentioned, this is an active process, so it is absolutely going to be using things like pumps and exchangers and co-transporters and antiporters, but there's also going to be a fair amount of passive transport that is going to occur here as well. So both active and passive transport is going to take place. When we talk about the passive transport, we are, of course, talking about things that are being driven by the concentration gradient. And when things are driven by the concentration gradient, basically we're talking about the difference in the values of those things from the concentration gradient. But remember, we also talked about just the overall amount of stuff that is in the blood as well. And remember, that sets up an osmolarity gradient. Right? We talked about this way back in 430 when we were talking about the tonicity. Right? We were talking about how when you're dehydrated, they don't just take deionized water and pump it into your body, because that upsets the osmolarity, the tonicity of your blood, and suddenly that blood rushes into your cells and causes it to burst. By the same token, if you're, shipwrecked at sea, you don't start drinking the ocean's water because of all the salt that's in there, right? That is a hypertonic solution, and it dehydrates your body. So osmolarity, the amount of stuff, plays an important role in that movement of water and movement of materials as well. Let's see some examples of this. And again, as we talked about, 25% of your blood is going to your kidneys at rest. And not surprisingly, about 20 to 25% of the oxygen being used by your body when you're at rest is to make ATP in your kidneys. So that shouldn't be too terribly surprising. Here we see this nice simple illustration of basically what we have here in our tubule, proximal convoluted tubule, most likely, simple cuboidal cell with microvilli, interstitial fluid, and then that paratubular capillary that is around it. I think we mentioned this, but if not, let me see if I do it here. Yep, there we go. There are tight junctions, and if you remember, tight junctions include these transmembrane proteins that attach from one cell to the next, forming these big, long web-like structures. Kind of looks a little bit like quilting or like a zipper that connects them together. These tight junctions help to serve two main functions. One of those main functions is it is going to limit, notice I didn't say stop, but limit the materials that can pass between the cells. And remember, our plasma membrane is a fluid mosaic. So by having these tight junctions, we can separate the proteins so that the proteins that are involved in transport on the apical surface are going to be different from the proteins that play a role in transport on the basal surface. So we are going to, as we saw, if you think back to digestion, when we have different routes for things going through the apical and the basal surface, it's the same type of thing here, right? Just like we saw in digestion, our materials, water and solutes, need to pass through two membranes. And as they pass through those two membranes, they are going to be able to get back into the body, right? Because remember, anything that stays in the tubule becomes urine and leaves the body. So these tight junctions are going to allow us to separate the materials, the proteins, the transporters that are on the apical surface and the basal surface. All right. Now, when we talk about the reabsorption routes, the most obvious one is the paracellular reabsorption. Paracellular reabsorption, as I mentioned, this gap in between allows for a limited, the tight junctions limit, but don't preclude entirely the movement of materials between the cells. And in fact, for certain ions, chloride happens to be a decent example of this. As many as 50% of those ions and some water can actually be reabsorbed paracellularly. Others, we want more control over. And those that we want more control over, they are going to have to go through the membranes, first the apical and then the basal. And much like we saw in the digestion, there are going to be different pathways for things across the apical and the basal surface. And just like in digestion, we will learn these reabsorption routes, and I guarantee you one of them will be an essay question on the exam. So we will talk about those transcellular reabsorption routes in a bit. All right, hold on, let me see here. Okay, perfect, that's what I want. All right, so most materials, and let's go ahead and draw this. Here is our absorptive cell with its microvilli. And most of the materials, as we talked about, are going to travel transmembrane through the apical and the basal surface. All right. So for something like a glucose to be able to get in, there is going to be some type of specific glucose transporter, especially here on the apical surface. And as we talked about, these molecule-specific proteins, these specific transporters, are vitally important for regulating the reabsorption of those resources. But the flip side of that is that they also limit the movement of the substances. Right? They typically move those substances in one direction, and they're going to limit the movement of them. Again, the way I always like to think of this is that if you are at the mall, for instance, right, you're at the Galleria and it has two floors, a top floor and a bottom floor. You cannot move unlimitedly between the top and the bottom floor. Right? You can't jump high enough to reach the top floor from anywhere you want. I guess technically you could jump off the top floor from anywhere you want, but you would definitely get hurt on the way down. You are limited in the number of ways you can get from the bottom of the mall to the top of the mall, right? Elevators, stairs, escalators, things along those lines. And there are a limited number of those, and they can only carry a limited amount of people. Right? When things are normally going fine at the mall, it's not usually an issue. But if suddenly BTS was doing a free concert on the second floor, everyone would be trying massing to that escalator to get up there. But would everybody necessarily be able to get on it at once? No, there's a limited capacity that it is able to carry. And the same thing is true for our transporters. Transporters have what we call a transport maximum. That is the maximal amount of whatever it is, sodium, potassium, glucose, whatever it is that they are able to carry. The nice thing about a transport maximum is if you know how much of a substance is in your blood, or really the plasma, then, and you know the transport maximum, you will know whether you will find it in the urine. Let's take a couple simple examples. The transport maximum for glucose is about 180 milligrams per deciliter. Does anybody know what a normal person's glucose level in their blood should be? I'll wait if you have to look it up on the interwebs. Come on, someone give me something. What is a normal, healthy glucose level in your blood? Well, here, we'll ask Siri then, since no one else seems to be able to come up. And Siri, what is a normal glucose level in a healthy person's blood? There you go. I like that. 140 is a good number. Excellent. Is that higher or lower than the maximum amount that our nephron is able to transport? These questions are getting easier. Is 140 higher than or less than 180? It's less than. Excellent. So as a result of that, in a normal, healthy individual, do you expect there to be glucose in their urine? No. Right? So again, all of the glucose should be reabsorbed, basically because of what's normal in the level and the transport level of this. About 100%, not about 100% of glucose in a normal, healthy individual will be reabsorbed. Now, if you happen to have uncontrolled diabetes mellitus, glucose in your blood could get as crazy high as 350 milligrams per deciliter. If that happens to be your glucose level in your blood, if it's uncontrolled, then are you going to see glucose in the urine? Yeah, absolutely. Right. In fact, that's one of the first tests that we do to look for diabetes. Right, is there urine, is there glucose in the blood? In fact, they've been doing this since ancient times. In medieval times, if you had a baby that was really crying and really upset, one of the first things the doctor would do is taste the urine. That's one of the reasons why doctors make so much money, is they have to taste all the urine. Right, and of course, in medieval times, what were they tasting the urine for? They were looking to see if the urine was sweet, to see if that glucose was available. Right? And everything has a different transport maximum. For amino acids, it's 65 milligrams per deciliter, and so on and so forth. The goal of this, then, this transport maximum, we can look at the levels in the blood, we can look at the transport mechanisms, maximums of our kidneys, and that will tell you whether or not that substance is going to be present in the urine. So if I give you the levels of it in the blood and I give you the transport maximum of it, you should be able to tell me whether it would be present in the urine or not. Right? Under normal conditions, glucose isn't. Here we see the pathway, and we'll talk about the pathway of glucose in the next class, but it's going to be reabsorbed. It comes back in through. And like I said, under normal healthy conditions, we absorb 100% of that glucose. But like I said, if you have uncontrolled diabetes mellitus, not all of that glucose is going to be reabsorbed. Right? We call this condition glucosuria, where there's glucose in the urine. And we can see two implications of this. I got this animation from a fellow instructor many years ago. I like it. Oh, it's not working. That's weird. Why are you not working? Oh, there it is. Excellent. So notice here in the case of glucosuria, there's far more glucose in the filtrate than our poor little transporters are able to bring back. So some of that glucose stays in the filtrate, some of it stays in the tubules and ends up in the urine. And one of the implications of having uncontrolled diabetes mellitus is with all that sugar staying in the kidneys, as we know, water likes to follow stuff. So a lot more water will stay in the filtrate as well. So typically someone who has uncontrolled diabetes mellitus, not only do they have sugar in their urine, but they typically produce a larger volume of more dilute urine because less water is able to be reabsorbed as a result of all that sugar in their blood and therefore in their filtrate. So this transport maximum is important for understanding what and how much of those things are going to be reabsorbed and determining what will be in the urine. All right, questions on that? All right, I know this is a little bit of a shorter lecture, but that is actually the things that I wanted to make sure we got covered today so that we aren't too far behind when we get to Wednesdays. So this is everything that I wanted to cover. It is a little bit of a shorter lecture, but it does allow us to cover the things and stay on track, which is the important thing. So again, those of you who did show up, I want to thank you greatly for this. And everybody else, and you guys included, I will see you guys on Wednesday, the 21st? Wednesday the 21st. So excellent. So any questions before we call it a day? All right, excellent. Then